Good morning, all of you. So, first of all, very happy Independence Day 2021. So, in this day, we have to salute the martyrs for their sacrifices and we have to thank them for our freedom. Okay, let's get into the topic. Today, we are going to see about the different forms of media. So, there are three different types of media. So, different forms: semi-solid form, liquid form, and solid form. In solid form, it was again categorized into the three types. So one is agar slope, another one is agar deep, and agar plate. Okay, we'll see. First one is liquid broth. So liquid broth, it doesn't contains any of the solidifying agent. So only the liquid particle is present. So it is mainly used for the culturing of organisms, in addition to used for the some of the tests to be taken care of. So for example, for the fermentation studies also we can use this organisms by means of the test. So that is called sugar fermentation test. In order to characterize the organisms, we are using the broth, that is the preliminary biochemical characterization by using the MRVP broth. Other than this also the different types of tests are there. So that is all are called liquid broth. So if we are not adding any of the solidifying agent, that is called as liquid broth. And for example is the nutrient broth. It does not contain any of the solidifying agent. So it is mainly used for the culturing of the organisms, and if you want large amount of organisms, any type of organisms we have to culture, it means we can go for the nutrient broth. In addition to that, if we want to enrich the nutrient broth with the help of some other additional nutrient supplements also, example blood, serum and sugars for any other special purposes. And solid media contains 1.5 to 2% of agar. So agar acts as a solidifying agent. It does not involve in any of the reactions. Mainly the solid media is used for the isolation of bacteria, and mainly in agar plate for isolating the individual organisms is done with the help of this type of solid media. And agar slope is after boiling that agar containing the media, it was poured into the test tube and it was taken by the slant position. After the agar cools and it was solidified, the tube is on the slant position, right? So the agar, it was in a slanting position like this. So in this we are sticking the culture, so we will get the separate segregate colonies as well as storing the organisms in this agar slant. And agar deep, and this is the one of the interesting technique. So it is used for the isolation of anaerobic organisms or the find out of anaerobic organisms. So in this we have to fill the test tube with the agar containing media. So it was created the oxygen gradient in the test tubes. So in the top layer the oxygen is available. If it is go into the deep, there is a depletion of oxygen. So we are using the fine needle to prick into that. So organisms grow in the upper surface, it is called aerobes. So if it is go into the deep, only the organisms grow in that deep part, that is called obligate anaerobe. Strictly doesn't need any of the oxygen. So in between gap the organisms grows, it is called facultative anaerobes. It can use some amount of oxygen, but some only grow in the top of the layer. So when it doesn't grow any of the inside, means that is called ablative aerobe. Okay, so semi-solid media. So come to semi-solid media, it is so in between that solid media as well as the liquid media. So in the liquid media we have to add the 0.5% of agar concentration or less. So whereas in case of the solid media we are adding that 1 to 1.5 or 2% of the agar. So we are adding in semi-solid media only 0.5%. So it is also used for the identification or the cultivation of the microaerophilic bacteria, and also it is used for the identification of the motility nature of the organisms, observing the movement of the organisms with the help of this semi-solid media. And it is a comparison of all these things. This is the solid one agar plate, and this is an agar deep, and this is an agar slant. Okay, you have to find the difference between these two, agar slant and agar deep, and this is the broth liquid one. Okay, so all these photos I have taken from the net, and if you want any clarifications, any doubts, you can mail me sivasa91@gmail.com. Don't hesitate to ping me. And some of the questions to think: what are the different forms of media, and what is the main use of semi-solid media, and what is called agar deep, and what is the use of that, and what is the main difference between the solid and the liquid media, by which component we are differing these two, and which form of media is used to isolate the anaerobes, and what is called anaerobe and aerobe. You have to be clear in all these concepts. Okay, if you know the answer, please comment in the comment section box. And thank you. And once again, happy Independence Day, all of you. Thank you.